Hi, I would just like to start my presentation by thanking the BNS for giving me the opportunity to present some of my research from my PhD, which is looking at the neuroinflammatory response in the motor regions in human sporadic motor neuron disease. While MND predominantly affects motor neurons, glial cells are also known to contribute to the progression of motor neuron disease. Activated microglia, the predominant immune cells of the central nervous system, are routinely observed as microgliosis at the key sites of neuronal degeneration. And the presence of activated microglia has been found to correlate with a number of key features of disease, including the extent of TDP-43 pathology, a key pathological hallmark of MND, as well as motor neuron loss and, importantly, the rate of disease progression. The main studies which have looked to characterize this neuroinflammatory response have primarily relied upon the use of animal models of familial MND, such as mutant SOD1 murine models. And to broadly summarize these studies, they highlight the importance of both neuroprotective and neurotoxic microglial responses over the course of disease. However, many of these studies have contradictory results and have not translated as well as hoped to human drug treatment trials. And this could be due to a number of reasons, including differences between murine and human microglia, and critically, most animal models represent familial MND, which accounts for only 10% of human motor neuron disease cases, while the majority of MND is sporadic in nature, and they show very different disease pathology and progression. As such, my study is a post-mortem study which aims to characterize the neuroinflammatory and microglial response in human MND. And we started by looking at neuroinflammatory gene expression changes. To do this, we extracted RNA from the ventral horn of spinal cord and grey matter of the motor cortex from two cohorts of MND and control cases and measured gene expression using the nanostring neuroinflammation panel. The results were then validated using publicly available datasets. The results of this gene expression analysis showed an increase in inflammation in the spinal cord in MND cases, with 100 genes being differentially expressed. The majority of these genes, 62, were upregulated. Key pathways were linked to specific inflammatory pathways, such as the tumour necrosis factor signalling pathway and the NF-kappa B pathway, as well as markers of phagocytosis and the ApoE and TREM2 signalling pathways. By contrast, motor cortex showed very little change in inflammatory signalling compared to control cases. And this indicates that potentially very different disease processes are taking place across different regions of the central nervous system in motor neuron disease. MND cases were taken forward for further analysis to identify those genes associated with the length of survival. Within the spinal cord, 23 genes were associated with more rapid decline, and these were associated with T cell recruitment as well as cell survival, myelin stabilization, cytoskeletal reorganization, and potassium buffering. 23 genes were also associated with a slower disease progression, and these were linked with cytokine and immune receptors, as well as the gene CD163 and DNA damage and repair response and autophagy. Interestingly, although the motor cortex was not associated with very much change when compared to control, a number of genes were associated with survival. 17 genes were associated with more rapid patient decline, and these were linked to T cell recruitment and pro-inflammatory cytokines. And 30 genes were associated with a slower disease progression, and these were associated with DNA repair and methylation, ubiquitination and autophagy, as well as toll-like receptor signaling, and the signaling pathways NF-kappa B and transforming growth factor beta. By comparing key genes and pathways from our spinal cord datasets, I've compiled some targets which are being taken forward for further analysis using immunohistochemistry. And the first of these targets is the TREM2 TYRO-BP ApoE signaling pathway. TREM2 and TYRO-BP form a signaling complex associated with the surface of macrophage cells, which increases the transcription of inflammatory molecules and can increase phagocytic and proliferative cell behavior. TREM2 has been found to be upregulated in motor neuron disease. ApoE, while more widely associated with Alzheimer's disease, has also been found to be upregulated in motor neuron disease, and a paper from 2017 found that ApoE was able to bind TREM2 complex, resulting in a switch in microglial phenotype from their homeostatic state to a more toxic neurodegenerative specific state, and we hope to explore this more in motor neuron disease. My next target is CD163, which is a haptoglobin hemoglobin receptor and particularly expressed by perivascular macrophages. However, it is also thought to be expressed by microglia following blood-brain barrier dysfunction. My next target is CTSS, which is part of the cathepsin family, and its upregulation is associated with pro-inflammatory signaling. CTSS is also downregulated by the expression of cystatin C, one of the primary components of Benina bodies of pathology associated with motor neuron disease. We will also be looking at key microglial markers such as IBA1 and HLA-DR, which are associated with microglial activation, and CD68, a marker of phagocytosis, as well as TDP43 as a marker of pathology. The next part of my project aims to examine the changes in the protein expression of those specific neuroinflammatory targets and to also look at the changes in microglial morphology. We have started staining in cervical spinal cord and most of our data analysis has been performed using Visiopharm software. Looking at the first of our microglial markers, CD68, we see a very clear shift from the classical ramified morphology, which is observed here in our control cases, to this much more amoeboid morphology observed in our MND cases. And we see a significant increase in the percentage of areas stained in the ventral horn and in the lateral corticospinal tract. Next, looking at MHC class 2 or HLA-DR, we see again a very clear switch in microglial morphology between our control and our MND cases, and a significant increase in staining in the lateral corticospinal tract. Next, looking at CD163, in our control cases, staining was mostly limited to the blood vessels and perivascular macrophages. However, in our MND cases, we see widespread staining across the spinal cord. A significant increase in staining was observed between control and MND cases in the ventral horn and in the lateral corticospinal tract, and this result could indicate blood-brain barrier dysfunction in MND. In tyrosine binding protein, we saw some glial staining in our control cases, however, this was much more profound in our MND cases, and we saw a significant increase of staining in the ventral horn. Looking at our ApoE cases, we see widespread glial staining in our control cases. However, in our MND cases, we see much more abundant staining and staining that looks to be astrocytic in nature as well. We see a significant increase in staining density in the lateral corticospinal tract. Finally, we have begun looking at the expression of TREM2. Expression of TREM2 has been routinely observed in mouse microglia, and therefore it was assumed that human microglia also express TREM2. However, it's been very difficult to confirm the expression of TREM2 in the human central nervous system using immunohistochemistry. Instead, staining tends to be limited to monocyte type cells and blood vessels unless there has been significant damage to the blood-brain barrier, which could indicate that expression of TREM2 observed in the central nervous system is the result of infiltrating peripheral immune cells. Therefore, we have started to look at the expression of TREM2 in spinal cord using a co-localization study. To do this, we first stain our cases with TREM2 using an alcohol-soluble dye and image our slide. We then decover slip the slide and dissolve the stain in alcohol, and then restain for a microglial marker. We then scan this slide, and by lining up the images, we are able to look at which cells are positive for both markers. So far, we have been able to observe a subpopulation of cells which are positive for both TREM2 and IBA1, as well as some cells which are only positive for our microglial markers. The cells positive for both TREM2 and IBA1, which is shown here by green arrows, show a very small and compact morphology, and there is no sign of ramified or amoeboid morphology associated with microglia, therefore indicating that these cells might not be microglial. However, we will continue to investigate this using other microglial markers. So to briefly summarise, we first looked at changes in the neuroinflammatory gene expression signature in the motor regions of the central nervous system and found the spinal cord to be a much more inflammatory environment than the motor cortex in motor neuron disease. And from these data, we have identified key pathways which we've taken forward for further analysis using immunohistochemistry. Currently, I'm working on finishing off the staining of those key targets in cervical spinal cord, and I'm also looking at performing semi-quantitative analysis for the slide stained with ApoE and TyroBP to look at details in the staining which could not be picked up using the Visiopharm software. I'm also looking at validating the TREM2 antibody and the expression of TREM2 by human microglia, and I'm expanding the IHC analysis to motor cortex and non-motor regions of the brain using TMA technology. I would just like to thank you all for listening and for all of those who have made this project possible. And thank you to the donors to the Sheffield Brain and Tissue Bank and their families. If there's any questions, I'm happy to answer them now. Thank you.